Hi, I'm Shannon Becker,
And I'm Dragone.
and today we're going to talk about the enzyme Monoamine Oxidase A. Monoamine Oxidase A, or MAO-A, is found in humans, primates, cats, rodents, and cows. It's in the enzyme class amine oxidase in the subclass flavonoenzyme. This means that it uses the cofactor flavonoadenine dinucleotide, or FAD. FAD is significant because it is covalently bound through thioether linkage to the enzyme and is the only redox cofactor required for the catalysis of the reaction. Related enzymes to MAO-A are MAO-B, which is the only other flavonoenzyme amine oxidase, as well as lysyl oxidase, primary amine oxidase, and diamine oxidase, which all have copper instead of FAD as their cofactor.
So next we're going to talk about the function and mechanism of the enzyme. The function of the enzyme is to break down monoamines in mammals. Monoamines consist of serotonin, dopamine, and norepinephrine. These are important because they're not only mood-regulating chemicals, but they also determine how we interact with our environment. The mechanism of the breakdown of the monoamines: when a FAD and a monoamine both bind to the enzyme, the FAD acts as a proton acceptor, which converts the monoamine into an imine. This imine is then able to be broken down through hydrolysis of water, which ends its biological significance. MAO-A can be inhibited by MAO-A inhibitors, which prevent its function and function as antidepressants for some patients. This is due to the resulting increase in monoamines as the enzyme is no longer allowed to break them down, and for some patients this provides a very nice increase in mood. Another change of MAO-A is something known as Brunner syndrome, which is named after the family that it was located in. This is a low genetic expression of the enzyme, which leads to increased impulsiveness, increased aggression, and a higher incidence of alcoholism alongside very low intelligence quotients. This is not it does not happen very often, but it's very bad when it does occur.
So on the gene level, MAO-A is located on the X chromosome at position P11.23. It has 15 exons. The main transcription factors are SP1, GATA2, and TBP. In terms of post-translational modification, it uses N-terminal acetylation of the methionine. Additionally, this is closely related to MAO-B, as we've already mentioned. MAO-B, instead of acting on serotonin, norepinephrine, and dopamine, works almost exclusively on dopamine. Additionally, MAO-B is located on an adjacent strand to MAO-A. So we've looked into the similarities between MAO-A and MAO-B using bioinformatics. Essentially what we did was we looked up the genome sequence of both MAO-A and MAO-B in humans, and then compared them using BLAST. As you can see here, the low E-value and the high identity score, as well as the high number of BLAST hits, 82, indicate the close relationship between the two.
So next we will move on to the structure of monoamine oxidase A. What we have here is called a ribbon model. It shows the entire molecule from the N-terminus at the top to the C-terminus at the bottom. This is separated into a dimer, where the top exists in the cytosol, or extracellular part. It consists of a yellow group and a red group, and the blue group is inserted into the outer mitochondrial membrane. The yellow portion of the ribbon is the FAD binding region, where FAD binds as a cofactor. There is a small black molecule in the middle of the yellow section that represents FAD. The red portion of the ribbon, on the other hand, represents the substrate or inhibitor binding section. Currently, there is a green molecule in there that represents a reversible inhibitor known as aramine hydrochloride, which is later utilized in our research experiment. The blue region at the bottom consists of a number of positively charged amino acids which form helical tails and attach itself into the outer mitochondrial membrane. It is a monomer until inserting into the mitochondrial membrane.
So the research article that we looked into was the structure of human monoamine oxidase A at 2.2 angstrom resolution, the control of opening the entry for substrates or inhibitors. This experiment took place in 2008 and was spearheaded by the Japanese scientist Dr. Sun. Now, the purpose of their experiment was to determine the important amino acids involved in substrate binding. They did this in order that it might be used for future medical applications, especially in the case of inhibitors, which, as we've mentioned, could play an important role, do play an important role in antidepressants. Additionally, this was done to get a better picture of the structure of MAOA, to also gain a better understanding of the function of MAOA, especially considering that up until this point, they had only had a low resolution structure of MAOA in rats. Now, they determined the structure by comparing a deliberately mutated strand of MAOA to wild type MAOA in both rats and humans.
So the first step was to construct expression plasmids for both the wild type and mutant MAOA. They began by taking the cDNA of human MAOA and amplifying it by PCR, which is the traditional method. To create the rat MAOA, they took the cDNA and inserted it into a yeast expression vector known as YEP51. And then finally, to create the mutated strand, they used a site-directed mutagenesis kit, which translated the 110th residue, which is normally glycine, into alanine. They did this to determine if there was any difference in function or structure, such as folding. To create their crystallized enzymes in order to perform X-ray diffraction experiments, they utilized a method known as the vapor drop method. This is an uncommon method of creating crystals. To perform the vapor drop method, they take a reagent and a sample. They have an elevated platform in a well in which they place a drop of the reagent and sample mixed together. The sample in this case is our enzyme. And then they also place the reagent into a well. The reagent seeks to maintain an equilibrium in pressure, which is depicted by the equation at the top. And by the end of this equilibrium being carried out, if performed correctly, in the raised platform, there will be a crystal remaining. So for this experiment, they crystallized the enzyme alongside aramine hydrochloride. This was the aforementioned reversible inhibitor. They did this so that they could find out if there was any difference in binding between the mutants and the wild type variants. They did this by mixing the protein and aramine at a ratio of 1 to 5. They determined that they could create their best crystals by having a reagent of 1.6 molar ammonium sulfate and 100 millimolar citric acid while performing the experiment at 277 degrees Kelvin. Post-crystallization, they froze the crystals in a nitrogen gas stream at 100 degrees Kelvin in preparation for their X-ray diffraction experiments. In their X-ray diffraction experiments, they took 5 wild-type crystals and 2 mutant crystals and performed X-ray experiments for data collection. Fortunately for the scientists, the wild-type diffracted isotropically into its phase diagram, which means that every single time they performed the experiment, it created the same image. So they were able to use that image. Unfortunately, for the mutant crystals, they diffracted anisotropically, which means that each phase diagram was slightly different and provided a slightly different resolution. So what they had to do is they took 232 images of varying resolutions and combined them into an overall image.
They then determined the structure by using molecular replacement method. This essentially means that they compared the wild type and mutated strands using the model of rat MAOA, given that the rat MAOA didn't contain the C terminals. In this model, the mutants had replaced at position 110, glycine with alanine. They did so to see if the change in structure would create any change in the substrate binding capabilities. Additionally, since this was at position 110, they were able to see if the flexibility of the fold also had an impact on substrate binding. In this version as well, they used human now A as a dimer, as a monomer instead of a dimer, which indicates that it was not bound to the mitochondrial membrane. Now, as a result of this, in the binding to the mitochondrial membrane, where this is the mitochondrial membrane and this is the cytosol, we find, again, the C-terminus here, and here we have many positively charged amino acids, namely arginine and lysine. These are important because they interact with the outer mitochondrial membrane. And here we have another depiction. In this case, they are again comparing human now A to human now B, as well as to rat now A. In this case, they were looking for the important amino acids, which are all indicated on the diagram as well as on our slide. On the diagram, the yellow indicates human now A, the blue human now B, and the orange rat now A. Now the slight differences in the structure between human now A, human now B, and rat now A accommodate the slight changes in function. In conclusion, they found the important residues that interact with the substrate, and they came up with the first high-resolution model of human now A.
So what are the future implications of this study? Well, perhaps with the increased knowledge on the important amino acids for substrate or inhibitor binding, they will be able to create better inhibitors that are more specific and may have a better antidepressant role. This also may be able to aid in the understanding of other amino oxidases, as they are currently very misunderstood molecules.
These are our sources.